Today we're going to begin Chapter 3, the cells, its functions, and cell division. First, there's an idea or a concept called the cell theory. It was developed by Robert Hooke's research in 1665. There are a couple things that you need to know. All cells are the building blocks of plants and animals. All cells come from the division of pre-existing cells, and cells are the smallest units that perform all the vital physiological functions. Lastly, each cell maintains homeostasis at a cellular level, and the human body contains trillions of cells. There are four major parts of the animal cell. 1, the cell membrane, 2, the cytoplasm, 3, the organelles, and 4, the nucleus. The two major types of cells in the human body are 1, sex cells or germ cells. They include the reproductive cells or male sperm, and lastly, female oocyte, a cell that develops into the egg. Secondly, somatic cells. Soma means body, so all body cells except the sex cells. The secondary division or secondary cells are prokaryotes. They're cells that don't have a nucleus, and eukaryotes are cells that do have a nucleus. In this image of the cell model, we see the plasma membrane, the non-membranous organelles, and the membranous organelles, and we're going to begin to go over those. In this particular image, we see the microvilli. They're membrane extensions that increase the surface area to facilitate the absorption of extracellular materials. We also can see the microfilaments. Here we see the cilia. They're long extensions containing microtubule doublets. We see the proteasomes. There are hollow cylinders of proteolytic enzymes with regulatory proteins at their ends. And we see the ribosomes, and there's two types. There's free ribosomes and fixed ribosomes, and they produce proteins. Here we see the Golgi apparatus, and it's stacks of flattened membranes, and its function is storage, alteration, and packaging of secretory products and lysosomal enzymes. We see the mitochondria, we see the endoplasmic reticulum, and we see both the rough ER and the smooth ER, and then we see the peroxisomes. Again, we see the peroxisomes here, and their function is catabolism of fats and other organic compounds, the neutralization of toxic materials generated in the process. And then the lysosomes, they contain digestive enzymes, and their function is to the removal of damaged organelles or pathogens. Here we have the nucleus, and in the middle we have the nucleolus with the nucleoplasm. Remember, the nucleus is the main control center of the cell, and it's where all genetic information is stored. Now let's go back to the cell membrane. The cell membrane, we have an extracellular fluid, ECF, and that's called interstitial fluid. It's a watery membrane that surrounds the cell. The cell membrane, plasma membrane, or plasmolemma, separates the cytoplasm from the extracellular fluid. Cytoplasm is also called cytosol. Cytosol is the liquid in the cytoplasm, and then the intracellular structures collectively are known as organelles. The plasma membrane has certain functions. The first function is physical isolation. It's a barrier. Secondly, it's regulation of exchange with the environment. It's a semi or selectively permeable membrane. It also is about 6 to 9 nanometers thin. The next function of the plasma membrane is it's sensitive to the environment. And the final function of the plasma membrane is it gives structural support. It anchors the cells and tissues. Some of the anatomy of the plasma membrane, it's made up of membrane lipids. It has a phospholipid bilayer. The phospholipid anatomy, it has hydrophilic heads, which means water-loving heads, and they exist towards the watery environment on both sides. In other words, the extracellular fluid, the ECF, and the intracellular fluid, the ICF. But it has hydrophobic tails or fatty tails, and that's inside the membrane. And it's a barrier to ions, water, and certain other substances. Within the plasma membrane, there are also membrane proteins. This is very important. There's integral proteins within the membrane and peripheral proteins bound to the inner or outer surface of the membrane. There are also receptor proteins that bind and respond to ligands, ions, or hormones. There are carrier proteins, and what they do is they transport specific solutes through the membrane, and then there's protein channels, and they regulate water flow and solutes through the membrane. This is an illustration of the plasma membrane. You can see it has a phospholipid bilayer. You can see the extracellular fluid or interstitial fluid outside the cell membrane, and you can see the cytoplasm inside the cell membrane. It's a fluid mosaic model, and you're able to see all the membrane proteins. Inside the cell, we have cytoplasm. That's all the materials inside the cell and outside the nucleus. Cytosol is the intracellular fluid. It has dissolved nutrients or dissolved materials in it. For example, nutrients, ions, protein, and waste products. It's very high in potassium and low in sodium. It has high in protein. It's also high in carbohydrate. However, it's low in amino acids and fat. And then the organelles are structures with specific functions, and they are within the cytoplasm, but outside the nucleus. There are two types of organelles. There are non-membranous organelles. They have no membrane, and they have direct contact with the cytosol. They include the cytoskeleton, microvilli, centrioles, cilia, ribosomes, and proteasomes. Know that there's a specific question on that. And then the membranous organelles, they're covered with a plasma membrane. They're isolated from the cytosol. They include the ER, the endoplasmic reticulum, the Golgi apparatus, the lysosomes, the peroxisomes, and the mitochondria. We are now going to talk about the non-membranous organelles. The 6 non-membranous organelles include cytoskeleton, microvilli, centrioles, cilia, ribosomes, and proteasomes. The cytoskeleton is important. It has structural proteins for strength and shape. It includes thick filaments, intermediate filaments, microtubules, and this makes up the cell's structure and size. First, let's go over the microvilli. Again, these are all the non-membranous organelles. They increase the surface area for absorption of different substances, and they attach to the cytoskeleton. The centrioles and the centrosome, they're very important in that they form the spindle apparatus during cell division, know that for the test, and the centrosome cytoplasm surrounding the centriole. The cilia are small hair-like extensions, and the cilia moves fluids across the cell surface. The ribosomes are a very important part of the cell. There are 2 types. There are free ribosomes and fixed ribosomes. The function of the ribosomes is to build polypeptides in protein synthesis. The free ribosomes are in the cytoplasm, and they manufacture proteins for the cell. The fixed ribosomes are attached to the ER and manufacture proteins for secretion. Lastly, the proteasomes contain enzymes called proteases, and they disassemble damaged proteins for recycling.